Hey everyone, welcome to the PowerPoint and lecture module for module 10.2.1. And here we'll focus on the autonomic nervous system, which is also known as the ANS, which will adjust your basic life support systems without your conscious control. In the next section we'll also consider aspects of higher order functions such as consciousness, learning, and intelligence. And then finally, within that stuff, we'll look at the effects of aging on the nervous system and conclude with an overview of interaction between the nervous system and other body systems. So to get started here, your conscious thoughts, plans, and actions are only a tiny fraction of what the nervous system actually does. If all consciousness were eliminated, your vital physiological processes would still continue virtually unchanged. After all, a night's sleep is really not a life-threatening event. Longer, deeper states of unconsciousness are not necessarily more dangerous as long as you get nourishment and other basic care. People with severe brain injuries can survive in a coma for decades. So how do people survive under those conditions? Their survival is possible because of the autonomic nervous system. So the figure on the previous slide compares the organization of the somatic and the autonomic nervous systems. Both are efferent divisions that carry motor commands. The SNS controls skeletal muscles, and the ANS controls visceral effectors such as smooth muscle, glands, cardiac muscles, and adipocytes. The primary structural difference between the two is that the SNS, motor neurons of the central nervous system, exert direct control over skeletal muscles. And in the ANS, by contrast, motor neurons of the central nervous system synapse on visceral motor neurons and the autonomic ganglia, and these ganglionic neurons control visceral effectors. The hypothalamus contains the integrative centers for autonomic activity, and the neurons in these centers are comparable to the upper motor neurons in the SNS. Visceral motor neurons in the brainstem and the spinal cord are known as the preganglionic neurons because they extend to the ganglia. These neurons are part of the visceral reflex arcs. Most of their activities represent direct reflex responses rather than responses to commands from the hypothalamus. The axons of preganglionic neurons are called preganglionic fibers. Preganglionic fibers leave the CNS and synapse on ganglionic neurons, visceral motor neurons in the peripheral ganglia. These ganglia, which control hundreds of thousands of ganglionic neurons, are called autonomic ganglia. Ganglionic neurons innervate visceral effectors such as smooth muscle, glands, cardiac muscles, and adipocytes. The axons of the ganglionic neuron are called postganglionic fibers because they begin at the autonomic ganglia and extend to the peripheral target organs. Somatic or visceral sensory information can trigger visceral reflexes, and the ANS distributes motor commands of those reflexes. Sometimes those motor commands control the activities of target organs. For example, in cold weather, the ANS stimulates the erector pili muscle and gives you goosebumps. In other cases, the motor commands may alter some ongoing activity. A sudden loud noise can startle you and make you jump, but thanks to the ANS, that sound can also increase your heart rate dramatically and temporarily stop all digestive gland secretion. And these changes in visceral activity take place in response to neurotransmitters released by postganglionic fibers. As noted earlier, a specific neurotransmitter may stimulate or inhibit activity depending on the response of the particular plasma membrane receptor. We consider the major types of receptors later on in these PowerPoints, but let's turn to the anatomy and physiology of the ANS at this point. So each autonomic ganglion functions somewhat like a baton handoff in a relay race. Within the ganglion, one runner, or the preganglionic fiber, hands off the baton, a neurotransmitter, to the next runner, which is the postganglionic fiber, who then continues on to the finish line, or the target effector. You're probably already familiar with the names of the two main subdivisions of the ANS, known as the sympathetic division and the parasympathetic division. And the two divisions work in several ways. One, most often these two divisions have opposing effects. If the sympathetic division causes excitation, the parasympathetic causes inhibition. The two divisions may also work independently. One division innervates some structure, and the two divisions may work together, with each controlling one stage of a complex process. In general, the sympathetic division kicks in only during exertion, stress, or emergency, and the parasympathetic division predominates under resting conditions. An increase in sympathetic activity prepares the body to deal with emergencies. So imagine walking down a long, dark alley and hearing some strange noise in the darkness ahead. Your body responds right away, and you become more alert and aware of your surroundings. Your metabolic rate rises quickly, up to about twice its resting level. Your digestive and urinary processes stop temporarily, and more blood flows from your skeletal muscles. You begin breathing more quickly and more deeply. Both your heart rate and blood pressure increase, circulating your blood more rapidly, and you feel warm and you begin to perspire. And we can summarize this general pattern of responses to increased levels of sympathetic activity as follows. 1. Heightened mental alertness. 2. Increased metabolic rate. 3. Reduced digestive and urinary functions. 4. Activation of energy reserves. 5. Increased respiratory rate and dilations of respiratory pathways. 6. Increased heart rate and blood pressure. And 7. The activation of sweat glands. The parasympathetic division stimulates visceral activity. For example, it brings about the state of rest and digest after you eat a big dinner. Your body relaxes, the energy demands are minimal, and both your heart rate and your blood pressure are relatively low. Meanwhile, your digestive organs are highly stimulated. The overall pattern of responses to increased parasympathetic activity are as follows. 1. Decreased metabolic rate. 2. Decreased heart rate and blood pressure. 3. Increased secretion by salivary and digestive glands. 4. Increased motility and blood flow to the digestive tract. And 5. Stimulation of urination and defecation. The ANS also includes a third division, the enteric nervous system, or the ENS. The ENS is an extensive network of neurons and nerve networks in the walls of the digestive tract. The sympathetic and parasympathetic divisions both influence the enteric nervous system, but many complex visceral reflexes are initiated and coordinated locally. They operate without instructions from the CNS. Altogether, the ENS has roughly 100 million neurons, at least as many as the spinal cord. It also uses the same neurotransmitters found in the brain. And throughout this kind of section, we'll focus on the sympathetic and parasympathetic divisions, which integrate and coordinate visceral functions throughout the body. This figure shows the overall organization of the sympathetic division of the ANS. Its preganglionic neurons are located between the segments T1 and L2 of the spinal cord, and its ganglionic neurons are located in the ganglia near the vertebral column. The cell bodies of the preganglionic neurons are in the lateral gray horns, and their axons enter the ventral roots of these segments. The ganglionic neurons are in 3 locations. 1. The sympathetic chain ganglia. And sympathetic chain ganglia are also called paravertebral ganglia, and they lie on both sides of the vertebral column. Neurons in these ganglia control effectors in the body walls, inside the thoracic cavity, and in the head and limbs. 2. The collateral ganglia, and those are also known as the prevertebral ganglia, and they're anterior to the vertebral bodies. Collateral ganglia contain ganglionic neurons that innervate tissues and organs in the abdominopelvic cavity. And 3. The adrenal medullae. The center of each adrenal gland is known as the adrenal medulla, or the suprarenal medulla. It is a modified sympathetic ganglion. The ganglionic neurons of the adrenal medullae have very short axons, and when stimulated, they release their neurotransmitters into the bloodstream, not at a synapse. This mode of release allows the neurotransmitter to function as a hormone, affecting target cells throughout the body. In the sympathetic division, the preganglionic fibers are relatively short because the ganglia are located near the spinal cord. In contrast, the postganglionic fibers are relatively long, except at those adrenal medullae. The ventral roots of the spinal segments T1 to L2 contain sympathetic preganglionic fibers that originate from the lateral gray horns. After passing through the intervertebral foramen, each ventral root gives rise to the myelinated white ramus, which carries myelinated preganglionic fibers into a nearby sympathetic chain ganglion. These fibers may synapse within the sympathetic chain ganglion and or at one of the collateral ganglion in the adrenal medullae. They diverge extensively, with one preganglionic fiber synapsing on 24 or more ganglionic neurons. Preganglionic fibers running between the sympathetic chain ganglion interconnect them, making the chain look like a string of pearls. Each ganglion in the sympathetic chain innervates a particular body segment or group of segments. If a preganglionic fiber carries motor commands targeting structures in the body wall, thoracic cavity, head, neck, or limbs, it synapses in one or more sympathetic chain ganglion. The unmyelinated postganglionic fibers then follow differentiating paths depending on where their targets lie. Postganglionic fibers that control visceral effectors in the body wall, the head, the neck, or the limbs enter the gray ramus and return to the spinal nerve for subsequent distribution. And these postganglionic fibers innervate sweat glands, smooth muscles, and superficial blood vessels, and erector pili muscles. Postganglionic fibers innervating structures in the thoracic cavity, such as the heart and the lungs, form bundles known as sympathetic nerves. This figure shows the sympathetic nerves on the left side and the spinal nerve distribution on the right, but in reality, both innervation patterns occur on either side of the body. The next figure provides a more detailed view of the structure of the ganglion chain and the sympathetic division as a whole. On the left side of the figure shows the distribution to the skin and to the skeletal muscles and the tissues of the body wall. The right side shows the innervation of visceral organs. Each sympathetic chain contains 3 cervical, 10 to 12 thoracic, 4 to 5 lumbar, and 4 to 5 sacral ganglia, plus 1 coccygeal ganglia. The numbers vary because adjacent ganglia sometimes will fuse. Preganglionic neurons are limited to spinal cord segments T1 to L2, and these spinal nerves have both white rami, myelinated preganglionic fibers, and gray rami, or unmyelinated postganglionic fibers. The neurons in the cervical, inferior lumbar, and sacral sympathetic chain ganglia are innervated by preganglionic fibers that run along the axis of the chain. And in turn, these chain ganglia provide postganglionic fibers through gray rami to the cervical, lumbar, and sacral spinal nerves. As a result, only spinal nerves T1 through L2 have white rami, and every spinal nerve has a gray ramus that carries sympathetic postganglionic fibers for distribution to the body wall. The spinal nerves provide somatic motor innervation to skeletal muscles of the body walls and limbs, but they also distribute sympathetic preganglionic fibers. About 8% of the axons in each spinal nerve are sympathetic postganglionic fibers. In the head and neck, sympathetic postganglionic fibers leaving the superior cervical sympathetic ganglia supply the regions of structures innervated by cranial nerves 3, 7, 9, and 10. So in summary, the cervical, inferior, lumbar, and sacral chain ganglia receive preganglionic fibers from spinal segments T1 through L2. Only the thoracic and superior lumbar ganglia, T1 through L2, receive preganglionic fibers from white rami, and every spinal nerve receives a gray ramus from a ganglion of the sympathetic chain. The abdominal pelvic viscera receives sympathetic innervation by sympathetic preganglionic fibers that synapse in separate collateral ganglia. These fibers pass through the sympathetic chain without synapsing. They form the splenic nerves, which lie on the posterior wall of the abdominal cavity. In adults, the collateral ganglia are typically single rather than paired, and they originate as paired ganglia, so left and right, but the two usually will eventually fuse. Postganglionic fibers leaving the collateral ganglia extend through the abdominal pelvic cavity, innervating a variety of visceral tissue and organs. The general function is to 1. Reduce blood flow and energy use by the organs that are not important to immediate survival, such as the digestive tract, and 2. Release stored energy. The splenic nerves innervate 3 collateral ganglia. Preganglionic fibers from the 7 inferior thoracic spinal segments at either end of the celiac ganglion or the superior mesenteric ganglia. These ganglia are embedded in an extensive network of autonomic nerves. Preganglionic fibers from the lumbar segments form splenic nerves that end in the inferior mesenteric ganglia. All 3 ganglia are named after nearby arteries. The celiac ganglion is named after the celiac trunk, which is a major artery, and its branches supplying the stomach, spleen, and liver. The celiac ganglia commonly consist of a pair of interconnected masses of gray matter located at the base of the artery. The celiac ganglia may also form a single mass or many small interwoven masses. Postganglionic fibers from this ganglia innervate the stomach, the liver, the gallbladder, the pancreas, and the spleen. The superior mesenteric ganglia is found near the base of the superior mesenteric artery, which provides blood to the stomach, the small intestines, and the pancreas. Postganglionic fibers leaving the superior mesenteric ganglia innervate the small intestine and the proximal 2/3 of the large intestine. The inferior mesenteric ganglia is located near the base of the inferior mesenteric artery, which supplies the large intestine and other organs in the inferior portion of the abdominal pelvic cavity. Postganglionic fibers from this ganglia provide sympathetic innervation to the kidney, the urinary bladder, the terminal portions of the large intestine, and the sex organs. Preganglionic fibers entering the adrenal gland proceed to its center, or a region called the adrenal medulla. The adrenal medulla is a modified sympathetic ganglion where preganglionic fibers synapse on neuroendocrine cells, which are specialized neurons that secrete hormones into the bloodstream. You'll get more into hormones as you get into AMP2, but for right now, if you don't know much about them, just think of them as chemical messengers here. These neuroendocrine cells secrete the neurotransmitters epinephrine, which we'll refer to as E, and norepinephrine, which can be referred to as NE. Epinephrine, or adrenaline, makes up about 75 to 80% of the secretory output. The rest is NE, or noradrenaline. The bloodstream carries the neurotransmitters throughout the body, where they cause changes in the metabolic activities of many different cells. The effects resemble those produced by the stimulation of sympathetic postganglionic fibers. They differ, however, in two respects. One, the cells are not innervated by sympathetic postganglionic fibers. And two, the effects last much longer than those produced by direct sympathetic innervation, because the hormones continue to diffuse out of the bloodstream for an extended period of time. The sympathetic division can change the activities of tissues and organs by releasing NE at peripheral synapses and by distributing E and NE throughout the body in the bloodstream. The visceral motor fibers that target specific effectors, such as smooth muscle fibers in the blood vessels of the skin, can be activated in reflexes that do not involve other visceral effectors. In a crisis, however, the entire division responds. In this event, called sympathetic activation, it's controlled by sympathetic centers in the hypothalamus. The effects are not limited to peripheral tissues, because sympathetic activation also alters CNS activity. When sympathetic activation occurs, a person experiences the following changes: increased alertness by stimulation of the reticular activating system, causing the individual to feel on edge, a feeling of energy and euphoria, often associated with disregard for danger and a temporary insensitive to pain stimuli, increased activity in the cardiovascular and respiratory centers of the pons and medulla oblongata, which increases blood pressure, heart rate, breathing rate, and depth of respiration. A general elevation in muscle tone through stimulation of the medial and lateral pathways so that the individual looks tense and may begin to shiver. The mobilization of energy reserves through the accelerated breakdown of glycogen in muscle and liver cells, and the release of lipids by adipose tissue. And these changes, plus the peripheral changes also noted, prepare the individual to cope with a stressful situation. So we've examined the structure of the sympathetic division of the ANS and in general effects of the sympathetic activation. Now let's consider the cellular basis of these effects on peripheral organs. When stimulated, sympathetic preganglionic neurons release ACH at synapse with ganglionic neurons. Synapses that use acetylcholine as a transmitter are called cholinergic. The effects of the ganglionic neuron is always excitatory. These ganglionic neurons then release neurotransmitters by specific target organs. The axon terminals differ from the neuromuscular junctions of the somatic nervous system, and in the sympathetic nervous system, the telodendron form a branching network, with each branch resembling a string of pearls. Each pearl is a swollen segment called a varicosity and is packed with neurotransmitter vesicles. Chains of varicosities pass along or near the surface of effector cells, and these cells have no specialized postsympathetic membrane, such as the motor end plate and skeletal muscle cells. Instead, membrane receptors are scattered across the surfaces of the target cell. Most sympathetic ganglionic neurons release norepinephrine at their varicosities. Neurons that release norepinephrine are called androgenic. A small but significant number of ganglionic neurons in the sympathetic division release ACH rather than norepinephrine. Varicosities releasing ACH are located in the body wall, the skin, the brain, and the skeletal muscles. The norepinephrine released by varicosities affects its target until reabsorbed or inactivated by enzymes. 50 to 80% of the NE is reabsorbed by varicosities and is either reused or broken down into the enzyme monoamine oxidase, or MAO. The rest of the NE diffuses out of the area or is broken down by the enzyme COMT, which is catechol-O-methyltransferase, in the surrounding tissues. In general, the effects of the NE on postsynaptic membrane last for a few seconds, significantly longer than 20 millisecond duration of ACH effects. As usual, the response of the target cells vary with the nature of their receptors. When the adrenal medullae release NE or E into the bloodstream, the effects last even longer because 1, the bloodstream does not contain MAO or COMT, and 2, most tissues contain a relatively low concentration of those enzymes. After the adrenal medullae are stimulated, tissue concentrations of norepinephrine and epinephrine throughout the body may remain elevated for as long as 30 seconds, and the effects may persist for several minutes. The effects of sympathetic stimulation are the result primarily from the interactions of norepinephrine and epinephrine with androgenic membrane receptors. There are two classes of these receptors, alpha receptors and beta receptors. In general, norepinephrine stimulates alpha receptors to a greater degree than it does beta receptors, and epinephrine stimulates both classes of receptors. For this reason, localized sympathetic activity involving the release of Ne at viscosities primarily affects nearby alpha receptors. By contrast, generalized sympathetic activation and the release of E by the adrenal medullae affect alpha and beta receptors throughout the body. Alpha receptors and beta receptors are G protein coupled receptors. As we saw in some of the previous chapters, the effects of stimulating such a receptor depend on the production of secondary messengers, intracellular intermediaries with verified functions. The stimulation of alpha receptors activates their associated G proteins on the cytoplasmic surface of the plasma membrane. And there are two types of alpha receptors. You have alpha 1 and alpha 2. Alpha 1 is the more common type of alpha receptor, and its stimulation results in the G protein activation that brings about the release of intracellular calcium ions into the cytosol from reserves in the endoplasmic reticulum. This action generally has an excitatory effect on the target cell. For example, the stimulation of A1 receptors on smooth muscle cells causes peripheral blood vessels to constrict and sphincters along the digestive tract and urinary bladder to close. Stimulation of A2 receptors and the G protein activation result in a lowering of cyclic AMP, or also known as cAMP, levels in the cytoplasm. Cyclic AMP is an important secondary messenger that can activate or inactivate key enzymes. This reduction generally has an inhibitory effect on the cell. The presence of A2 receptors in the parasympathetic division helps coordinate sympathetic and parasympathetic activities. When the sympathetic division is active, the norepinephrine release binds to A2 receptors at parasympathetic neuromuscular and neuroglandular junctions and inhibits their activity. Beta receptors are located on plasma membranes of cells in many organs, including skeletal muscles, the lungs, the heart, and the liver. The stimulation of beta receptors and G protein activation triggers changes in the metabolic activity of that target cell. These changes occur indirectly as G protein activation results in an increase in intercellular CAMP levels. There are three major types of beta receptors: beta 1, 2, and 3. The stimulation of beta 1 receptors leads to an increase in metabolic activity. For example, the stimulation of B1 receptors in skeletal muscles alters or accelerates the metabolic activities of the muscles. The stimulation of B1 receptors in the heart increases the heart rate and forces contraction. The stimulation of B2 receptors causes inhibition, triggering and relaxation of smooth muscles along the respiratory tract. As a result, respiratory passageways dilate, making breathing easier. The inhalers used to treat asthma trigger this response. A third type of beta receptor, or B3, is found in adipose tissue. Stimulation of B3 receptors leads to lipolysis, or the breakdown of triglycerides stored within adipocytes. The fatty acids generated through lipolysis are released into the bloodstream for use by other tissues. The activity of these synapses produces vasodilation and increased blood flow through these regions. So to summarize, 1. The sympathetic division of the ANS includes two sets of sympathetic chain ganglia, one on each side of the vertebral column, 3 collateral ganglia anterior to the vertebral column, and 2 adrenal medullae. 2. The preganglionic fibers are short because the ganglion are close to the spinal cord. The postganglionic fibers are longer and extend a considerable distance to their target organs. 3. The sympathetic division allows extensive divergence. That is, a single preganglionic fiber may innervate 24 or more ganglionic neurons in different ganglia. And as a result, single sympathetic motor neuron in the CNS can control a variety of visceral effectors and can produce a complex and coordinated response. 4. All preganglionic neurons release ACH at their synapses with ganglionic neurons. Most postganglionic fibers release NE, but a few release ACH or NO. The effector response depends on the second messenger triggered by G protein activation when Ne or E binds to alpha receptors or beta receptors. So the parasympathetic division of the ANS consists of 1. Preganglionic neurons in the brainstem and the sacral segments of the spinal cord. The midbrain, the pons, the medulla oblongata contain autonomic nuclei associated with cranial nerves 3, 7, 9, and 10. In the sacral segments of the spinal cord, the parasympathetic nuclei lie lateral to the gray horns of spinal segments S2 through S4. 2. Ganglionic neurons in peripheral ganglia within or adjacent to target organs. Preganglionic fibers of the parasympathetic division do not diverge as extensively as those of the sympathetic division. A typical preganglionic fiber synapse is on 6 to 8 ganglionic neurons. All are in the same ganglion. The postganglionic fibers then extend to the same target organ. The ganglion may be a terminal ganglion located near the target organ, or an intramural, or murus, meaning wall, ganglion, embedded in the tissues of the target organ. Note that this pattern makes the effects of parasympathetic stimulation very specific and localized. Terminal ganglion are usually paired. Examples include the parasympathetic ganglion associated with the cranial nerves. Intramural ganglion typically consist of interconnected masses and clusters of ganglion cells. Parasympathetic preganglionic fibers leave the brain and cranial nerves 3, 7, 9, and 10. These fibers carry the cranial parasympathetic output to visceral structures in the head. These fibers synapse in the ciliary, submandibular, and the aortic ganglion. Short postganglionic fibers continue to their peripheral targets. The vagus nerve alone provides roughly 75% of all parasympathetic outflow. It supplies preganglionic and parasympathetic innervation to structures in the neck and the thoracic and abdominopelvic cavities, as distant as the distal portions of the large intestine. The many branches of the vagus nerve mingle with preganglionic and postganglionic fibers of the sympathetic division, forming plexus comparable to those formed by the spinal nerves innervating the limbs. And we consider these plexuses in a later section. The preganglionic fibers in the sacral segments of the spinal cord carry the sacral parasympathetic output. These fibers do not join the ventral roots of the spinal nerves. Instead, they form distinct pelvic nerves, which innervate intramural ganglion in the walls of the kidney, the urinary bladder, the terminal portions of the large intestine, and the sex organs. The functions of parasympathetic division center on relaxation, food processing, and nutrient reabsorption. This division has been called the anabolic system because its stimulation leads to a general increase in the nutrient content of the blood. In response to this increase, cells throughout the body absorb nutrients and use them to support growth and cell division to create energy reserves in the form of lipids or glycogen. The major effects of the parasympathetic division include the following: constriction of the pupils to restrict the amount of light that enters the eyes, and focusing of the lenses of the eyes on nearby objects. Secretion of digestive glands, including salivary glands, gastric glands, duodenal glands, intestinal glands, the pancreas, so exocrine and endocrine, and the liver. The secretion of hormones that promote the absorption and use of nutrients by peripheral cells, changes in blood flow and glandular activity associated with sexual arousal, an increase in smooth muscle activity along the digestive tract, the stimulation and coordination of defecation, contraction of the urinary bladder during urination, constriction of the respiratory passageways, and a reduction in heart rate and forced contraction. All parasympathetic neurons release ACH as a neurotransmitter. The effects on the postsynaptic cell can vary widely, however, due to different types of receptors or to the nature of the second messenger involved. The neuromuscular, so neuron to muscle, and neuroglandular, or neuron to effector, junctions of the parasympathetic division have narrow synaptic clefts. The effects of stimulation are short-lived. Most of the ACH released is inactivated at synapse by acetylcholinesterase. Any ACH diffusing into the surrounding tissue is then activated by the enzyme tissue cholinesterase. As a result, the effects of parasympathetic stimulation are quite localized, and they only last a few seconds. The parasympathetic division uses the same transmitter, ACH, at all of its synapses, so neuron to neuron, and neuromuscular or neuroglandular junctions. Two types of ACH receptors occur on the postsynaptic membranes. You have one, the nicotinic receptors that occur on the ganglion cells of both parasympathetic and sympathetic divisions. They also occur at the neuromuscular junctions of the somatic nervous system. ACH always causes excitation of the ganglionic neuron of the muscle fiber. It works by opening chemically gated sodium channels in the postsynaptic membrane. 2. Muscarinic receptors occur at cholinergic neuromuscular or neuroglandular junctions in the parasympathetic division. They also occur at the few of the cholinergic junctions of the sympathetic division. Muscarinic receptors are G-protein coupled receptors. Their stimulation and G-protein activation produces longer lasting effects than does the stimulation of the nicotinic receptors. The response can be excitatory or inhibitory depending on the activation or inactivation of specific enzymes. The names nicotinic and muscarinic originated with the researchers that found that dangerous environmental toxins bind to these receptor sites. Nicotinic receptors bind nicotine, a powerful toxin that's obtained from a variety of sources, including tobacco leaves. The highest nicotine levels of tobacco are about 3 milligrams per gram. And muscarinic receptors are stimulated by muscarinin, a toxin produced by some poisonous mushrooms. These toxins have completely separate actions. Nicotine targets the autonomic ganglia and skeletal neuromuscular junctions. Muscarinin acts at the parasympathetic neuromuscular or neuroglandular junctions, and they produce dangerously exaggerated, uncontrolled responses due to an abnormal stimulation of cholinergic or androgenic receptors. Nicotine poisoning occurs as little as 50 milligrams of the compound is ingested or absorbed through the skin. The signs reflect widespread autonomic activation. They include vomiting, diarrhea, high blood pressure, rapid heart rate, sweating, and profuse salivation. Convulsions occur because the neuromuscular junction of the somatic nervous system are stimulated. In severe cases, the stimulation of nicotinic receptors inside the CNS can lead to coma and death within minutes. The signs and symptoms of muscarine poisoning are almost entirely restricted to the parasympathetic division. They include salivation, nausea, vomiting, diarrhea, constriction of respiratory passageway, low blood pressure, and an abnormally slow heart rate, or bradycardia. Sweating, a sympathetic response, is also important here. For this reason, Amanita muscarin, commonly known as the flyageric mushroom, is used in some Native American sweat lodge rituals. This table compares key structural features of the sympathetic and parasympathetic divisions of the ANS. The differences in structure lead to differences in functions. The sympathetic division has widespread impact reaching organs and tissues throughout the body. The parasympathetic division innervates only visceral structures that are supplied by the cranial nerves or lie within the abdominal pelvic cavity. Some organs are innervated by just one division, but most vital organs receive dual innervation, so they receive instructions from both the sympathetic and the parasympathetic divisions. Where dual innervation exists, the two divisions commonly have opposing effects. Dual innervations with opposite effects is more obvious in the digestive tract, the heart, the lungs, and at other sites, the responses may be separate or complementary. And the next table will provide a functional comparison of the two divisions, noting the effects of sympathetic or parasympathetic activity on specific organs and systems. I would encourage you to pause here if you need to a little bit more at this table. Parasympathetic postganglionic fibers from the ciliary, the pterygoidopine, submandibular, and otic ganglia of the head travel by the cranial nerves to their peripheral destinations. Sympathetic innervation reaches the same structures by traveling directly from the superior cervical ganglia of the sympathetic chain. In the thoracic and abdominal pelvic cavities, the sympathetic postganglionic fibers mingle with parasympathetic preganglionic fibers, forming a series of nerve networks collectively called amniotic plexuses. They include the cardiac plexus, the pulmonary plexus, the esophageal plexus, the celiac plexus, the inferior mesenteric plexus, and the hypogastric plexus. Nerves leaving these networks travel with the blood vessels and the lymphatic vessels that supply visceral organs. Autonomic fibers entering the thoracic cavity intersect at the cardiac plexus and the pulmonary plexus. These plexuses contain sympathetic and parasympathetic fibers to the heart and lungs, respectively, as well as the parasympathetic ganglion whose output affects these organs. The esophageal plexus contains descending branches of the vagus nerves and the splenic nerves, leaving the sympathetic chain on either side. The parasympathetic preganglionic fibers of the vagus nerve enter the abdominal pelvic cavity with the esophagus. There, the fiber enters the celiac plexus, also known as the solar plexus. The celiac plexus and the associated smaller plexuses, such as the inferior mesenteric plexus, innervate viscera within the abdominal cavity. The hypogastric plexus innervates the digestive, urinary, and reproductive organs of the pelvic cavity. This plexus contains the parasympathetic outflow of the pelvic nerves, sympathetic postganglionic fibers from the inferior mesenteric ganglia, and the splenic nerves from the sacral sympathetic chain. Even without stimuli, autonomic motor neurons show a resting level of spontaneous activity. This background level of activity determines an individual's autonomic tone. Autonomic tone is an important aspect of ANS function, just as muscle tone is a key aspect of SNS function. If a nerve is absolutely inactive under normal conditions, then all it can do is increase its activity on demand. But if the nerve maintains a background level of activity, then it can increase or decrease its activity, providing a greater range of controlled options. Autonomic tone is significant where dual innervation occurs, and the two ANS divisions have opposing effects. It is even more important where the dual innervation does not occur. To demonstrate how autonomic tone affects ANS function, let's consider one example of each situation. The heart receives dual innervation, and recall that the heart consists of cardiac muscle tissue and specialized pacemaker cells that trigger its contractions. The two autonomic divisions have opposing effects on heart functions. Acetylcholine released by postganglionic fibers of the parasympathetic division causes a reduction in heart rate. Norepinephrine released by varicosities of the sympathetic division accelerates heart rate. Small amounts of both of these neurotransmitters are released continuously, creating autonomic tone. However, the parasympathetic division dominates under resting conditions. Heart rate can be controlled very precisely to meet the demands of active tissues through small adjustments in the balance between the parasympathetic stimulation and the sympathetic stimulation. In a crisis, stimulation of the sympathetic innervation and inhibition of the parasympathetic innervation accelerate the heart rate to the maximum extent possible. The sympathetic controlled blood vessels diameter demonstrates how autonomic tone allows fine adjustment of peripheral activities in target organs that are innervated by only one ANS division. Blood flow to specific organs must be controlled to meet tissue demand for oxygen and nutrients. When a blood vessel dilates, blood flow through it increases, and when it constricts, the blood flow is reduced. Sympathetic postganglionic fibers release NE at the smooth muscle cells in the walls of the peripheral vessels. This background sympathetic tone keeps these muscles partially contracted. So the blood vessels are ordinarily at about half their maximum diameter. When increased blood flow is needed, the rate of NE release decreases and the sympathetic cholinergic fibers are stimulated. As a result, the smooth muscle cells relax, the vessels dilate, and the blood flow increases. By adjusting sympathetic tone and the activity of cholinergic fibers, the sympathetic division can exert precise control of vessel diameter over its entire range. Recall that the centers involved in somatic motor control are found in all portions of the CNS. The lowest level of regulatory control consists of the lower motor neurons involved in cranial and spinal reflex arcs. The highest level of control consists of the pyramidal motor neurons of the primary motor cortex, operating with feedback from the cerebellum and the basal nuclei. Similarly, the ANS is also organized into a series of interacting levels. At the bottom are the visceral motor neurons in the lower brainstem and the spinal cord that are involved in cranial and spinal visceral reflexes. Visceral reflexes are autonomic reflexes initiated by the viscera. They provide autonomic motor responses that can be modified, facilitated, or inhibited by higher centers, especially those of the hypothalamus. For example, when a light is shown in one of your eyes, a visceral reflex constricts the pupils of both eyes, the consensual light reflex. The visceral motor commands are distributed by parasympathetic fibers. In darkness, your pupils dilate, but this pupillary reflex is directed by sympathetic fibers. However, the motor nuclei directing pupillary constriction or dilation are also controlled by the hypothalamic centers concerned with emotional states. For example, when you're queasy or nauseated, your pupils constrict. When you're sexually aroused, your pupils will dilate. Each visceral reflex arc consists of a receptor, a sensory neuron, a processing center, or one or more interneurons, and two visceral motor neurons. All visceral reflexes are polysynaptic. They can be long reflexes or short reflexes. Visceral sensory neurons deliver information to the CNS. It travels along the dorsal roots of the spinal nerves, within the sensory branches of the cranial nerves, and within the autonomic nerves that innervate visceral effectors. The processing steps involve interneurons within the CNS. The ANS then carries the motor commands to the appropriate visceral effectors. Long reflexes typically coordinate the activities of an entire organ. Short reflexes bypass the CNS entirely, and they involve sensory neurons and interneurons whose cell bodies lie in autonomic ganglia. These interneuron synapses on ganglionic nerves and then postganglionic fibers distribute the motor commands. Short reflexes control very simple motor responses with localized effects. In general, short reflexes may control patterns of activity in one small portion of a target organ. In most organs, long reflexes are most important in regulating visceral activities, but this is not the case with digestive tract and its associated glands. In these areas, short reflexes provide most of the control and coordination for normal functioning. The ganglia in the walls of the digestive tract contain the cell bodies of visceral sensory neurons, interneurons, and visceral motor neurons, and all of their axons form extensive nerve nets. Parasympathetic innervation by the visceral motor neurons can stimulate and coordinate various digestive activities, but the enteric nervous system is quite capable of controlling digestive functions independent of the central nervous system. Notice that the parasympathetic division participates in a variety of reflexes that affect individual organs and systems. This specialization reflects its relatively specific and restricted patterns of innervation. In contrast, fewer sympathetic reflexes exist. The sympathetic division is typically activated as a whole. One reason is that it has a high degree of divergence. Another reason is that the release of hormones by the adrenal medullae produces widespread peripheral effects. Centers in the brainstem that regulate specific visceral functions control the levels of activity in the sympathetic and parasympathetic divisions of the ANS. As in the SNS, in the ANS, simple reflexes based in the spinal cord provide rapid and autonomic responses to stimuli. Processing centers in the medulla oblongata coordinate more complex sympathetic and parasympathetic reflexes. In addition to the cardiovascular and respiratory centers, the medulla oblongata contains centers and nuclei involved with salivation, swallowing, digestive secretions, peristalsis, and urinary function. These centers are in turn subject to regulation by the hypothalamus. The term autonomic was originally applied because the regulatory centers involved with the control of visceral function were thought to operate autonomously, that is, independent of other CNS activities. This view has been drastically revised in the light of later research, but because the hypothalamus interacts with all other portions of the brain, activity in the limbic system, and other things, we now know that it's not autonomic.